Thought it might hit a few, you know, every little bit helps, so.
Yep.
So. Can't hear you, Joan. Cat got your tongue, Joan? You're halfway there, technical. Sign language.
You only get half credit. You only get half a makeup, John.
Tony, what do us old timers, how do we contribute to the?
You cut out, Joan, or Doug. How do you contribute to the what, Doug?
I don't do the web.
I don't understand your question. I heard you say, how do we contribute? And I didn't hear it after that.
You said that even though we're old timers, that we count somehow in the attendance.
No. I was teasing Joan because she's on visually, but we couldn't hear her. So I said she only got half a makeup for that, but I was just kidding.
Okay.
But if you're asking about rule of 85, I mean, rule of 85s get credit when they show up and they count. And if they don't show up, it doesn't hurt us. If that
Joan, if you go to the carrot top by the microphone down at the bottom, you can take a look and hit audio settings, and then it'll pop up a window where you may be able to test your microphone or something, or switch it around. So.
So Paul, what is that view behind you?
That is my timeshare in Silverthorne, Colorado.
I kind of suspected that.
Yeah.
It had that look about it.
Yeah. They're supposed to get 2 feet of snow in Denver this weekend.
I heard about that.
Yeah.
We had 18 inches of 18-inch snowfall on December 17. We've had 2 or 3 1 to 2 inch snowfall since then, and nothing else.
Are you still in Maine, Rich?
I am, Tony. Yeah. Going to be here for the duration. I think we'll probably come back maybe in January.
Wow.
Yeah.
Rich, it's good to see you. Hi, Norm.
Yeah.
Oh,
Frank.
Frank.
Yeah.
Morning.
Good to see Jeff Hamstra.
Hi, everyone.
hey, Jeff.
Hey, there he is, Mr. Hamstra.
Looks like Bob's back in Tucson.
I am. Hi.
Good trip.
It was. Thank you. We went out to California and it was great. How are you, Doug?
Doing better.
Attaboy.
So Bob, Rich Henry,
Hi, Rich.
you been playing golf?
Yes, every Wednesday.
Good for you. Good for you.
Next Wednesday, 9:30, Fred Enke, if you're interested.
Thanks. From 3,000 miles away, I don't think I can make it.
What the hell, a heck of a drive.
The next time for me will be mid-May, I think.
Rich, are you still in Maine?
I am, Frank. Yeah. Yep.
Did I tuned
Maine
in
has
late.
not loosened up too much yet, so we're still hanging in.
Have you got much snow up there yet?
Well, I was just telling folks, we had 18 inches in December on one snowfall and nothing of much appreciation after that. So it's been pretty meager for a Maine winter, I'll tell you that. Not that I'm disappointed. It's sunny and 57 or 58 right now.
Does Sharon get a 1.5 attendance points?
Yes, yes, I do.
Sure.
We're recruiting young these days.
Hi, Sharon, yeah.
Rich, I think you're going to be warmer in Maine this weekend than we're going to be in Tucson.
I have a friend that's been telling me about that in Tucson. He just went to California, but this morning at 7 o'clock when I woke up, it was 52 degrees here. I think it was in the high 30s in Tucson or something like that this morning.
Hey Sharon, can you tell us whether or not there's still tickets available for the ITK fundraiser next weekend?
For March 20th, there are still tickets available.
How do you
And-
get them?
You can go online to the ITK site, or you can email me and I'll send it to you directly. It's for the whole family this year. And there's a petition. So it should be a lot of fun. Liz is knocking at my door. I gotta go answer. Ford's in charge.
Oh, good.
Youngest member award.
He's subbing for Liz.
Dean McDonald.
Subbing for Sharon. He's looking at us like,
A
who are
member of Kinderact.
Hey, Joan, you might not know, but your camera's on. Well,
But her audio's not.
She's self-muting.
Okay, it's 10 after the hour. I'm going to go ahead and get started. Thank you everyone for showing up. Remember that Jeff Nordenson has the power of recording and then he can selectively edit. So just take that into account. I'm going to share my screen and we'll do the pledge. And then I believe Norm will be following up
There's salad too,
afterward
Liz.
with our
Lovely.
let me get this stuff here. Okay, come on, there we go. There we go.
I pledge allegiance to the flag of the United States of America and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all.
Stop that. Okay, I'm unmuted. If you would unmute. Okay, Norm, you're ready to go.
Please join me in a moment of reflection. Creator and sustainer of all that is or ever will be, accept our thanks for this day and all its blessings. We ask that you guide and direct our club, its leaders, and our actions. Grant that each of us may feel our responsibility to Rotary, to our community, to our country, and indeed to all countries and peoples. Bless our fellowship today in your service. Amen.
Thank you, Norm. Just wanted to let you welcome guests. We'll go through that in a moment. I sent out an email or a notice that I got from the county yesterday. It opened up for 55 and older and some contact stuff. So if anybody can take advantage of that, please do as I say, if I see stuff, I'll send it on to the club members. So hopefully that we can continue down that road to getting better. Andy, while you got timing's everything, just like at the dinner tables, could you kind of give us a little bit of an update on the funding that we have for Erickson's that we've collected and so on? And you may want to mention a little bit about the books. We don't have a full information on that, but just so the club gets an update.
Thank you, Paul, President Paul. Absolutely. When we set the stipend at $1,500 for Erickson, our goal was to make it a meaningful impact for the student. We have since learned that $1,600 would cover in-state tuition for this intern course, the 3 credit hours. And so the board at its recent meeting elected and approved a motion that we raise the stipend to $1,600. If we wind up with an out-of-state student, we'll cross that bridge when we come to it. Also, at the end of last week, I received from Kevra Rich the wish list book list for the library for Erickson School. And so we are going to be, I'm going to be trying to see if I can put prices against those books, and we'll see what we can do and start figuring out how to get information out to the club, to those who would like to participate either in purchasing books or in funding the purchase of books. So stay tuned. Thank you very much.
Thank you for that. Now I'll go back to the rest of my list. I got to read out my list. I'd like to welcome Dr. Betancourt. He is going to be our speaker here in a few moments from the U of A, and we'll learn some more information about COVID. It's good to see Mr. Hamstra taking a break from all those furnace calls. It's just about time to get ready for the AC calls. I saw, let's see, I say Merle's here. So hopefully it's, you'll probably get that storm in a little while. It's hitting the Rockies now. Good to see Pat coming in and here, and let's see. Oh, and everybody take notice. There's somebody, probably Rich is probably the furthest attendee here from Maine. He's joined us today, the wonders and wonderful world of Zoom. So good to see Rich from back way east. Let's see who else we got here. We got Arturo. Good to see you. Good to see you there on the screen. And I think I got to bring, and Joan is uh-oh, it shows like she's got a speaker turned off. So maybe she's, well, you may hear from her later. We'll see. So, but glad to see everybody. I'll go back down on my list a little bit then. Al, could you give the club just a couple, oh, look, and there's somebody actually in your screen to the side there. A little bit of a tidbit. Did you get that, Tony? Okay. Could you give us a little bit of an update on just where we are on fund giving? Just so the club kind of gives a tidbit of that. Just kind of same you gave to the board.
Yeah, which of course is not in front of me at this moment. So why don't you call on me a little later?
Okay, we'll come back. Don't forget to remind me because I'll forget too.
Okay.
Then the other thing, like I did probably right now, we had a board meeting on Tuesday. We discussed the bit with the Viscount. We're going to hold the dues like they were this quarter and not look to go back to the Viscount quite yet, but we will. I'm going to send out a Survey Monkey survey here probably tomorrow. Similar questions to before as far as vaccination, going back and things like that, so we can kind of get a pulse of the membership on how everybody feels with what's going to happen going forward, so we can maybe do a little bit more planning, maybe May, June, July, sometime in that time frame, so we can get an idea what people's thoughts are. Last week, if you remember, we took a little bit of a poll, and I think like 17 or 18 of the 25 people that responded had actually gotten their shots and so on like that. So maybe we're making a little bit of progress to get that taken care of, and maybe we'll be able to get back together sooner rather than later. So that'll be coming. And then finally, we're also in preparation for getting back into more of a virtual and in-person meeting looking for anybody that has maybe a little bit of technical knowledge as far as video and Wi-Fi type stuff, because we're probably going to have a camera or two and a speaker and all that, so we can look to stream things. That's going to be one of the big issues also with the Viscount. They're going to have enough bandwidth to be able to support things. So if anybody has some background and has some interest in kind of playing around and working on that, please let me know. Bob Grady's got a camera that he's kind of going to donate to the club to help out with a little bit of that. But other people that have some of that technical knowledge or have some things, it'd be nice to know. So that's where that's at. Al, did you find numbers?
Yeah, I've got it now. Thanks. What I shared with the board the other day were just some rough numbers of where we stand at this time in the Rotary year. And it's wonderful. Our annual fund goal for the year, people aren't going to remember that from report to report, so I'll keep reminding you, it was 12,005, which we thought at the time was a little bit low, but it's better to, overshoot than not, I guess. And anyway, we are now at 14, actually more like 15 because there's money in the mail. So goal was 12.5 and the current annual contribution total is over 15,000. So once again, Catalina's done a super job there.
Yay!
That is right. That is right. And we'll probably get double that when Anita gives us the 15 grand she's going to give us. Oh, okay. Well, what else do we have? The district is running at about $250 per capita. Oops, wrong. It's running at about $100 per capita right now, and Catalina Rotary is at 250 per capita, far exceeding the individual donor amount for the district. If you keep track of that sort of thing. That's probably it in rough numbers. I reported a couple of meetings ago, and I'll do it again right now, that one of the real points of pride for our Rotary Club this year is that we have exceeded, for the life of our club, the life to date of our club, we have exceeded $1 million in donations to the foundation. I don't know if there's very many clubs that can report that. So it's a thing to be proud of. Thank you, President Paul.
Okay, thank you for that. I see Tom Robertson's with us today. Good to see Tom. I saw actually a couple other folks. Dick Carlson, good to see you. Allison, see you on there. Love your background. And hopefully your roads are a lot better up there these days. So, and Jimmy's on board too. So with that, I thought that Matt was going to introduce our speaker, but I don't see that he's here. So I will go ahead and I'd like to introduce our speaker, Dr. Walter Betancourt. He's an environmental virologist and assistant research professor at the U of A. He earned his PhD in marine science from the University of South Florida. His most recent efforts have been focused on detection and identification of waterborne viral pathogens. Say that fast 3 times with a mouthful of food. And viral surrogates in water reuse systems for potable and non-potable applications. He's done a fair amount of research into this COVID stuff, getting and testing what's going on relative to where the things are, where they're showing hits, if you will. I asked him, I had the opportunity to ask him, where does he do the testing? He does that on the influence stuff. This is lunchtime, so I won't go any further than what that all means. But I will turn it over to Dr. Betancourt, and I'm anxious to listen to this. Having worked in the wastewater field for about 10 years, to hear what all's going on with what he's been doing and kind of how he's learned and what he's been able to find out about COVID and this going on. So, Dr. Betancourt, glad to hear you, and I'm really interested to hear what you've figured out and learned and such. So, all yours.
Thank you. Thank you, President, for inviting me to present to everyone of the Catalina Rotary, our surveillance program at the West Center. This is part of initiative that Dr. Ian Pepper, the West Center director, once we have the pandemic declared, So we were kind of limited in conducting our basic research. So then we said, well, we can conduct COVID-19 research because we have the capabilities. And then the first thing that we needed to do was developing the methods, because this is a novel virus, and this is obviously a group of viruses that are enveloped, and I'm going to talk a little bit about that later. But one thing that I want to make clear here is that it was kind of difficult for me to avoid any technical words, and because this is basically biology. So then it's going to be a little, let's say, slow in one way, and then another one, I have 21 slides, but I really appreciate this invitation, the opportunity, and I want to make it easy for everyone to understand, and then understand the principles of wastewater-based epidemiology to determine COVID-19 incidence in the population. So I'm going to share my screen here. Let me see if I can do that right now. If I am able to share.
There you go. You're heading that direction. There you go.
Okay, so let me see. Okay, I think I'm sharing my email.
PowerPoint stuff, you just have to make it full screen or make it the.
Yeah,
run
let me
it.
see. Okay, share here, share screen one, share. Okay, let me see if I can you see the PowerPoint or not?
We can see the PowerPoint. If you go down and just hit the little flag, yeah, the start, yeah, there, that one, that should do it for you.
Oh,
there
okay,
we go.
perfect. Okay, apologies.
It's got the preview slide, but we can still see what's going on.
Okay, perfect. So yes, the first slide that I'd like to show here is, this is an example of what viruses are. First of all, viruses are intracellular parasites, means that they need a cell in order to replicate. We have here poliovirus, we have adenovirus, so these are different viruses, reovirus. Major feature of these viruses, these are human viruses, so they infect humans, they cause disease in humans. They are naked, so they don't have any lipid layer like the SARS coronavirus. So here you can see that shapes is basically icosahedral. The size of these viruses is basically 30 to 50 nanometers. They can be RNA viruses, they can be DNA viruses. This here is a peppermint multivirus. This is a cylindrical virus and is infecting plants. We have seen that this virus is a good indicator of wastewater treatment performance, basically because this virus is found in high concentrations in sewage. So when we want to determine the reduction of viruses to the wastewater treatment process, we look at those viruses that we know that are going to be in high concentrations in sewage, because then we can determine log reduction through the wastewater treatment process. And if we are taking into account that advanced wastewater treatment process like membrane, integrated membranes like ultrafiltration and reverse osmosis, at that point, we are looking for those indicators that allows us to determine what is the efficiency of removal of that particular process. Now, as you remember, in 2019, December, so there was an emerging virus. It's called an emerging virus because it's a new virus that actually was, what we suspect is that the virus came from an animal and then jumped into humans. And so those are zoonotic pathogens. So pathogens or viruses that are present in animals, they don't cause disease in animals, but once they are, they have another host that can make the virus suitable for replication in humans. And that was the case with the SARS coronavirus 2. As you might remember, there was a SARS coronavirus 1 that was the same. The civets in China were actually the original host. And in this case, we don't know yet exactly what the host of this virus was initially, but actually originated in Wuhan, China. So then when we talk about parental strains, as you may know, there are new strains, variants, like the UK strain, Brazilian strain, South African strain, that they evolved from the Wuhan strain. So we have a parental strain, which is the Wuhan strain, and we have these new variants. They differ in many aspects, but let's go back to this. So the SARS coronavirus is in this slide here. This is a National Institute for Allergy and Infectious Diseases slide. And then what it's showing is that if you can see here, you can see probably the next one. I can show you here better. This is a depiction of what the SARS coronavirus is. And here you can see that the virus is actually present a lipid layer that it takes from the host. You know, remember our cells have a bilipid layer. So when the virus is present, is replicating in the cells. So they say this is the virus. It takes the receptor. It's a specific receptor present in the cells. His material is actually contain the information that the cells, the human cells, are gonna. He's gonna use the human cells in order to determine to request the synthesis of those components that at the end are gonna produce more viruses. Going back to the slide here, we can see that the virus is actually, this is the spike protein. The spike protein is a major component of the virus that binds to the receptors in the human cells. So this is necessary. So this is really specific. So human viruses has a specific receptors on the cells. They can infect human cells. Some animal viruses cannot infect, obviously, humans because we don't have the receptor for those. Now, these zoonotic viruses, what happens is that while they are transmitted among animals, they develop the capability to transmit to humans and then to be transmitted to humans. So what happens is that when we are in close association with these animals, then they jump the barrier, species barrier, and they can cause disease in humans. That's what happened with the SARS coronavirus 2. In this slide here, you can see that we have, this is a scanning electron micrograph. Viruses cannot be seen with the naked eye or with a regular microscope. So we need a magnification of the electron microscope, which is 50 or 500,000 X, which represent millions of times of magnification to be able to resolve, you know, the size of the virus, which is nanometers. SARS coronavirus is 160 nanometers inside, and then polyomavirus, for instance, is at 30 nanometers. So SARS coronavirus is kind of a large virus. It's an envelope virus too. So I explained that what happened is that the virus needs the cells, and then there is specific receptors, and this spike protein here is the one that binds to the cell receptor in the human cells. So we talk about, you know, the virus needs, it's an intracellular parasite because it needs the replication, it needs the cell in order to sequester the cell machinery in order to produce those components that are going to be assembled into a virion here. So when the virus exit the cell, he actually, he doesn't kill the cell, but he just take part of the membrane and it goes out. And you can see here, you see the spike proteins, and the blue component here represents the lipid, the bilayer lipid of the cell, of the human cell. So then this is the virion. So this is the virion that is infectious, and that is what is transmitted to other individuals. Basically, the virus is a respiratory virus. Now, we know that there are some individuals who has receptors also in the intestinal tract. So then initially we thought this is a respiratory virus, but then we found that the virus was present in sewage. So remember, you know, any respiratory problem, people tend to swallow, you know, mucus because they are inadvertently mucus, right? So then we think, okay, the virus is going to be excreted because it comes from the respiratory system. But actually, there were initial studies in February or January of 2020 where they the studies demonstrated that the virus actually replicated in the gastrointestinal tract. It can be excreted in feces by a group of individuals. So that was really important because then people working in the environmental virology field, we say, well, if we can do that with poliovirus, we have been done for years, actually part of the global polio eradication initiative from the World Health Organization, funded by Melinda and Bill Gates Foundation and other funding agencies. So their concern is that obviously poliovirus, there is a vaccine. So it doesn't make sense that at this time, 21st century, these diseases still occur. But what happened is that, particularly with poliovirus, in order to cover the amount of population in developing countries, I was initially explaining that they use an attenuated vaccine. The virus actually is kind of alive. So the virus replicate and produce the immunity that is necessary for individuals, once exposed to the poliovirus or the wild poliovirus, to be able to respond and then not get sick. But what happened is that the virus, when it replicates, let's say if we have an undercover population, let's say some people, and then remember for religious reasons, some people, they don't want to get vaccinated because they think that that's something else. So then, or people who are not vaccinated because of other wars or many issues that happen in the world and countries that have a lot of problems. So what happened is that the virus replicate more efficiently, and then the vaccine, the attenuated virus, can return to the neurovirulent strain. So now, instead of the wild poliovirus, which is still present in Pakistan and Africa, so the vaccine is actually causing the disease, and they call it vaccine-derived poliovirus. Now, just a background, because that's part of the environmental surveillance on poliovirus. So we transferred that to SARS coronavirus, knowing that the virus was actually excreted in feces. So then we know that the virus can be excreted at numbers of 10 to the 7, 10 to the 8. Now it makes sense that, a specific population is excreting these large numbers. When we go and then we test the sewage, we expect to find the genetic material. And that's something that I want to make clear later. But at this point, I want to show this slide. And then this is an RNA virus. It's a single stranded RNA virus. So this is the genetic material, similar that we have, let's say we have a genetic material that says, okay, you have green eyes, you have red hair. So this is part of the genetic material, the DNA, right? So these are viruses that are RNA. So it means that the RNA, if you remember about genetics, so is the DNA is converted to RNA, and from RNA to protein. So we need proteins because, this is our cellular components for membranes, for our tissues, general tissues of our hair, everything. So these are actually structural components, right? For the virus, it's actually the RNA that is actually synthesizing a specific, what we call structural proteins, like the nucleocapsid protein or the envolture protein, or other components of the virus, like non-structural proteins that are actually going to add, synthesizing components that are necessary for the virion, for the synthesis of the virion. So in this particular slide, what I'd like to show you is that we use those sequences in order to detect the virus using molecular techniques. Viruses can be cultured in cell lines that we can propagate in the laboratory. But to make it faster and to be able to detect viruses that probably we don't know that the virus exists, we can explore other possibilities like molecular assays. In molecular assays, we use specific primers, to amplify the genetic material. And then actually before it was like a single, very targeted approach to amplify genetic material, specific viruses. Nowadays, we have the capability to amplify, it's called virome. So it's next generation sequencing. We can identify a group of different viruses. And actually we are using these techniques nowadays to determine variations of the SARS coronavirus, because we can get the full genome, or at least full sequences of those target sequences of the interest that allows us to determine what is happening with the virus. So these, particularly the CDC, the Center for Disease Control, developed specific primers and a specific probe for detection of the virus using molecular techniques. And then against the nucleocapsid protein, it makes sense to do that because these viruses, they tend to evolve. So then what they did, they selected specific sequences that they are conserved, and then to be able to detect virus for long term. Now we need to take that into account because we know nowadays that variations can occur. So probably these targets are going to be for a while, but later we might need to change those targets. So, but this is really interesting in terms of what a depiction of the genetic material of the virus, and then how the virus used this, how the genetic material, once it's injected into the cells, can be used to produce these virion. So now this is really critical, because if I explain, when I explained you before that we have this receptor here we have the spike protein here that was the original one that was able to bind to the receptor in the cell, the human cell. So nowadays, what happened is that this spike protein, especially this one here, I mean, well, all the spike proteins of the virus, they have mutations. So when the virus replicates in the population, and that's what is critical nowadays, we keep our mask, we follow CDC guidelines. We also have the University of Arizona guidelines. So we need to stop, the reproduction rate of the virus. And then initially it was really high. The reproduction rate is actually what it's telling you is that you have a reproduction, it's usually called 1, 0, or greater than 1. So if you have a reproduction rate of a virus that is 1, it means that reproduction rate is actually how contagious an organism is, or the disease. Obviously, it's caused by the organism, right? So if we have a 1 R0 or reproduction rate, we know that one individual infected with that virus can cause disease in another individual. So if we have something that is less than 1, we know, oh, the virus is declining. So then the infection capability of the virus is declining. And that's what's happening with the Wuhan or the parental strain. But the new strains are taking over. So during the pandemic, the pandemic, when we were not aware that we have to take these precautions in order to avoid contagious or to be to contract the disease or to be exposed to people, one critical component here is that when the person is sick, you have still 5 or 6 days, you don't have any symptoms, but you are excreting a lot of viruses. So the first 5 days prior to the symptoms, the person is excreting a lot of viruses. So that's why we are unaware of that. And then if we don't wear masks, initially there were obviously lockdowns, there was not a vaccine, and then there was a lot of disease initially, and then obviously the hospital were overcrowded with people who were dying also of the disease because of that. So then we realized that there are symptomatic individuals. Now with this is different. Now we have mutations, particularly this one, the N501Y, which is basically was discovered in the UK. That's what they call it a lineage. Lineage means, or if I'm pronouncing that well, lineage means a strain that came from the original strain. Let's say if we talk about the father was the Wuhan strain that was originated in China. After that, when the World Health Organization declared the pandemic, obviously because it was already in every country. So then what happened is that the virus start replicating, many times in the population and they start mutating. So then in the UK, I think it's in October, November, they started conducting sequencing studies are going to be critical from now on until I don't know how long, but this is going to be very critical. So they discovered that were mutations, particularly this N501Y. This one is really important because it enhances the virus binding affinity to the cell receptor. It means that before it was okay, the virus was binding to the receptor in one way, and then probably the immune system was able to cope with that. Nowadays, the association is so high that, and I mean so high, but if I say it's a high affinity, because that's going to be the spike protein in the receptor, that's the term used for this purpose. So it's so high that then the contagious, the infectivity, I would say not the infectivity, the transmissibility is higher. So there is another mutation. There are 17 mutations actually in the lineage B117, which is the UK. And as you may know, there are now viral strains in California. There are viral strains in Washington, in the US, in many areas. Now, the B117 is already in the US and in many countries. So this was like the parental viral strain that originated and then has been propagated all over the world. So the second mutation, which is the 6917, is a deletion. So it makes this, particularly immunocompromised individuals, which is really critical because they say we have immunocompromised individuals where the virus find a way to replicate. So the virus is still replicating and it's creating more mutations, right? So we want to avoid that. Now, I am fully vaccinated. I got 2 doses of the Moderna vaccine, and then 1 month ago. So I am fully, let's say, immune. That's what I think, okay? My colleague in Israel, he's a retired professor from Tel Aviv University. He was really smart. He's a biologist. What he did was he got vaccinated with the Pfizer vaccine. So he took samples of blood, his own blood, and he tracked down the antibody titer. So initially, before he was not exposed to that particular virus, like everyone here in the US, we try to minimize exposure. We locked down, so he was in his house. So what he did was he tested his blood initially. And so he said, well, I don't have any antibodies at this point. Now he got vaccinated. The first time he got vaccinated, the levels of antibodies in his blood was 0.80. He got the second vaccine, and 2 weeks after that, the titer went to 5.83. Means that he was capable to produce a response against the virus. Now, I feel the same way, and then I guess many people who have get vaccinated and then are not sick, obviously are also full immune to the virus. So based on the antibody. Now, we don't know yet. We expect that these mutations and then the spike protein that has mutated, the vaccines, Moderna and then Pfizer, are effective against this mutation. Now, if we have a full immune system, we have been vaccinated, we are protected with our mask. Obviously, we are creating the barriers to minimize the high reproduction rate of either the new variants or the parental variant that is still present in the population, and then we can decrease that reproduction number. When we decrease that reproduction number, obviously we can go to 0. We can say, oh, we have a fully immunized population. And it's actually what is the trend in the US is that what's happening now. So we are creating herd immunity. People are getting vaccinated. People, for some reasons, are not getting vaccinated. But more people vaccinated means more immunity, herd immunity, and then we can destroy the virus, basically. But these mutations are really critical. So we still keep an eye on them because obviously if the transmissibility increases based on these mutations, we need to be aware of that because like I say, we don't know yet if it's going to be effective. The vaccines are going to be effective. Apparently, yes. And I've been in touch with colleagues who are actually physicians. They got vaccinated and they say, yes, we are going to be protected by this vaccine. But remember, this is a novel virus. And at this point, there are many things that we are discovering about coronavirus. Now, at the 21st century, we have the capabilities to follow this. And it's really important that, even after 1 year of the pandemic was declared by the WHO, now yesterday, we have, 6 months before that, we have, a vaccine already available. That didn't happen before. So that's really a blessing for the world and for the population. Now, these are things that I wanted to cover with respect to the biology of the virus. What viruses are, what SARS coronavirus is, what is the not physiology, but I would say the structure of the virus, how the virus transmits, and the sequences or the genome of the virus, why the virus is an RNA virus, how it replicates in the cells, and now the mutation. So this is the most updated information with respect to the situation of the virus in the pandemic era. Now, going back to what we work at the University of Arizona West Center, we started in March 2020, we started conducting surveillance in wastewater. So we use wastewater to monitor the incidence of COVID-19 in a community. We also, obviously it's important, if the virus is excreted in the feces and it goes to the influent, it's important to know if the virus is actually removed by wastewater treatment processes. And in fact, the virus is efficiently removed. Viruses, naked viruses like poliovirus, adenovirus, reovirus that I was talking before, naked, I mean they don't have an envelope, lipid bilayer. So the non-envelope viruses are more resistant, so they can be transmitted to the environment. Now, envelope viruses like coronavirus, Ebola, they are envelope and obviously they are weaker. in the environment, once the bilipid layer is destroyed, the virus is destroyed, basically. Now, what happened is that, to me, and then someone might say, well, I don't find this like a fat, but to me, the more pathogenic an organism is, the less survival in the environment, because the organism need to be transmitted among humans, to keep, the virus is really intelligent. It's just a protein code with nucleic acid, but he wants to perpetuate in the population because actually he wants his genetic material he wants to evolve, right? So then with respect to this, what we think is that if we eliminate the virus from the wastewater treatment processes, obviously sanitation is going to be key there. Now, the virus has one thing, like I say, it's very weak, but because it has this lipid layer, it can associate well with solids in suspension. So basically, primary treatment like sedimentation is really key at eliminating the virus. So we have tested secondary and tertiary effluent, and we didn't find the virus. Now, we found that the virus, chlorination is really important. One thing that I want to make clear here is that when we are detecting the virus in the environment, we are detecting the genetic material. At this point, we don't know yet if the virus is able to survive in sewage because we are detecting the genetic material. Makes sense to say, yes, we are detecting a viable and infectious viruses because we are detecting the genetic material. What happened is that RNA is really weak in the environment too. So we are dealing with a virus that is weak with a bilipid layer, and we are dealing with a genetic material that is also weak. Now, when we think, oh, we are detecting the RNA, we might think, okay, so means that the RNA of the virus is protected by the capsid, and the virus is infectious at that point. But if we don't have cell culture, if we don't have any idea of the viability, and that's what we are working right now, some confidential research and some research that requires biosafety level 3 capabilities. When I'm talking about surveillance, obviously this is done at biosafety level 2. What is a biosafety level 2? Biosafety, there are 3 levels of biosafety when you're working in a laboratory with pathogenic organisms. Biosafety level 1 means organisms that they don't cause disease in humans. Obviously, you have to follow guidelines in the laboratory. You cannot pipette with your mouth. You have to wear gloves. You have to wear a lab coat. You have to wear goggles, safety goggles or glasses in the laboratory. Biosafety level 2, now we have organisms that are pathogenic to humans. So we have to have a biosafety level 2 cabinet. So we have to work in a cabinet where we have pressure, right? Because we are dealing with something that is we have to have a barrier because we can get exposed to those organisms and get infected. But immunocompetent individuals, we have been exposed to so many of these organisms. We have also vaccines against polio, against other organisms, even against influenza. So then if we are working with biosafety level 2 organisms, we have to follow guidelines. We can do this with the SARS coronavirus 2 because we are working with sewage, but we have to follow additional precautions, additional measurements in order to work in the laboratory with SARS coronavirus 2 when we are dealing with wastewater. So obviously sewage has other microbial pathogens, so we have to work under those biosafety level 2 guidelines. Now for biosafety level 3, that's different. So you have to have protective equipment that requires specific training also, and then you have to be covered from head to toe. You have to have a respiratory device, so then you are not exposed to the environment that you are working on. And the laboratory is obviously sealed. I mean, obviously you have to enter the laboratory and go out, but anything that goes into the laboratory cannot go out. So it have to be there, and then have to everything to kill the virus have to be inside. So biosafety level 2 is a higher level of precaution in order to avoid exposure to pathogens that are really dangerous to humans. So at the West Center, we have developed the capability to conduct the surveillance. We are not only determining the removal by wastewater treatment processes that we already know that the virus is efficiently removed by wastewater treatment process to a point, right? Bioaerosol monitoring is another approach that we are dealing with. because we know, because it's a respiratory virus, we wanted to know if the virus in sewage treatment facilities who have the workers there present, so they are exposed to these sewage aerosols. So we wanted to know if there is aerosolization of these virions or viral particles. The other project that we are working on, and obviously this is initiative of the University of Arizona COVID-19 task force led by Dr. Ian Pepper at the West Center. It was an initiative also from colleagues of the University of Arizona, the president, Dr. Carmona, and other epidemiologists at the university, because we wanted this university first to reopen the university under safety conditions. And secondly, to ensure that obviously that the students and the staff are well protected. So nowadays we have obviously the vaccine, but initially when we initiated the surveillance was really key, because we wanted to know, using the principle of wastewater-based epidemiology, to determine the incidence of the virus in the sewage dormitories, because obviously the student population there are going to be excreting viruses. And the university took what is called test, trace, and treat. That was part of the mission of the university. So testing individuals coming to classes, those individuals who were infected, they were isolated or sent back home. Those individuals that were tested positive were dismissed or they were isolated. Those that tested negative were allowed to go to the dormitories. But obviously the surveillance was important at that point, because obviously there was interaction of the students, and then obviously at that time there was the pandemic was still, and then still is going on. Obviously it was critical to determine what was happening in the sewage, and obviously as a reflection of what's happening with the COVID-19. So the other point that we are working on is the assessment of SARS coronavirus 2 viability and persistence in sewage using in vitro cell culture. We're going to start doing this. We have the capability at the BioPhi Institute. We have 2 colleagues actually trained to work on the biosafety level 3 guidelines, and we have the blessing of the research laboratory safety system at the university to work biosafety under biosafety level 3 laboratory capabilities to determine viability of SARS coronavirus. So initially in March, when we started the surveillance, we selected wastewater treatment facilities across the US. And then we were also contacted by many countries, including Canada. They wanted to do also this type of surveillance, knowing that we advertised this in our website. And then we said, well, we are able to detect SARS coronavirus. We developed the methods. We have the capabilities to conduct these assays. And then we were contacted by this facility. So we asked them to provide information, what the population served was, the type of treatment that they provided. So then we have an idea of what we were dealing with. You know, if it was a large population, it was a small population, if the sewage was treated to what level. So that was important, because some facilities actually requested, we want to know if the virus is reduced. Some facilities, they say, we want to know if the virus is present in our community. So that was key at that point. So the first thing that we needed to do was developing methods for concentrating the virus, because first of all, this is an envelope virus. We were not dealing with this virus. We thought, oh yeah, the envelope viruses are really weak. They are not going to survive. Now, when we found that the virus was actually present excreted in the feces, present in sewage, we said, oh, we need to do something about this. So we developed the method. The method is really simple, but it's really, really specific and sensitive. Basically, the one here that I'm showing, number 1, we take the sewage, 500 mils or 1 liter, but out of those 500 mils, we just take 70 mils. And we do membrane filtration. What we do is that we use different membrane pore sizes, 0.8, 0.65, 0.45, and 0.22 to clean the water, because sewage has a lot of debris. So we want to get rid of the solids. We want to have the virus well concentrated or clean, or at least a suspension. Then we use what is called a centrifugal ultrafiltration. It's a device that has a very, very small pore size. Why we want that? Because we want to concentrate those particles efficiently. So then we do centrifugation, and the final concentrate is actually used for nucleic acid extraction. As I said before, we are detecting the genome genetic material of the virus. For this, we can use what is called RT-qPCR. I'm going to explain briefly what is that, digital PCR or droplet digital PCR. So when we started this in March, we saw what the principle of wastewater-based epidemiology was, and the feasibility of using the signals of the virus in sewage to determine incidence of the disease in the population. In this graph, you can see the bars here, the blue bars, indicate the concentration of the virus in genome copies per liter in sewage. Now, when we look at these straight lines here, the lines, the peaks here, this is the population sick, the clinical cases occurring in the population. 7 days prior to the disease, this is cumulative cases, and 7 days after the person initiated the symptoms. Obviously, these are based on Pima County Health Department data because they were doing the testing, the clinical testing. We don't do clinical testing, so but we use this in order to determine what was happening. So if you can see here, you can see from March, April, so there was an increase in the virus occurrence in sewage. And then there was also an increase in the incidence of the population, of the disease in the population. Now, this here, which is similar trend, they're just indicating that it's either the incidence of the disease either prior, 7 days prior to the symptoms or 7 days post-symptoms. Now, this is really important here because as you remember, so we say March, April, May, that's when the disease was kind of really high. Now, there was a lockdown imposed in, I think it was April, May, in Arizona. And you can see that during this time, the incidence of the SARS coronavirus in sewage was really low. So if we were talking before about 10 to the 6 genome copies per liter, at this point, we were talking about 10 to the 2, 10 to the 1, 10 to the 3 genome copies per liter. So very low levels. Now, in June, when the lockdown was relapsed, we can see an increase in this. So then wastewater-based epidemiology was capable of revealing the relationship between the occurrence of the virus in sewage and the incidence of the disease in the population. So this has maintained, and then nowadays we are actually, because we are co-located with the Agua Nueva Water Reclamation Facility, we have samples of sewage that we can process weekly. And then we are looking at the, first, the incidence of the disease is still kind of high, because we look at these 10 to the 6, but nowadays, remember, we probably have a higher level of immunity, might be asymptomatic individuals too, or might be new variants that are, what is called resurgence of COVID-19. So that's what is really critical to maintain, these precautions, wearing masks, even if you are fully vaccinated. So this is with respect to wastewater-based epidemiology and how wastewater-based epidemiology was able to reveal that relationship. So in order to continue with our research, we are exploring also aerosol samples. So we go to wastewater treatment facilities, and then we deploy these. This is a podcast. So this device has here is a membrane. So we capture all the air coming through this filter here. The filter is taken back to the laboratory, and we use a solution to elute or to wash what has been captured on the membrane, and then proceed with the isolation of the DNA and the detection. So now, how can we detect the virus? If we are talking about RNA, obviously the genome material, you might be familiar with what is called polymerase chain reaction. What we do is that we use the genetic material, we create the conditions in order to amplify these materials several times, and at the end we can quantify or we can determine if the virus was present there or not. And then we can also use this sequence to determine, oh, this is SARS coronavirus and this is the strain that is present in this community. So it's kind of a fingerprint if you look at that way. So it's like we are using the genetic material to detect the virus and also to identify the virus. Now, for this, the molecular assay that the Center for Disease Control developed was a reverse transcription quantitative polymerase chain reaction assay. This is something that is really, you can determine in real time based on this increase in fluorescence. Is telling you that there was an accumulation of the product during these amplification cycles. And the shorter the cycle, they say we have 5, 10, 15, 20 copies or cycles, cycles, in the fluorescent, the shorter the cycle number means more genetic material was present. So what we do first is we create what is called a standard curve. So we use a positive control, a non-sequence of the virus that we can synthesize these chemically. And then we use this to create different concentrations of the virus. And then we know that at cycle 5, we have, for instance, 10 to the 6. At cycle 10, we have 10 to the 5. At cycle 4, we have 10 to the 3, and so on. So then we create, that's a standard curve. Now, when we challenge our samples, then we're going to determine, that's what we call an unknown. So we're going to determine what the concentration of our unknown in our sample is with respect to what is in the standard curve. And then we can say, well, based on this concentration or this cycle number, this is the concentration corresponding to this standard. So then we have this concentration in this sample. So then what's what we call a relative quantification. But also we have the capability to conduct what is called droplet digital PCR or digital PCR, and that's an absolute quantification, meaning that if when we use our RT-PCR, we are using 20 microliters reactions. We have our components to determine if the genetic material was present or not. Now, when we do droplet digital PCR, we have the 20 microliters that is partitioned into 20,000 droplets, meaning that you have individual droplets with all the components to produce reactions. That is really key, because if you have a volume for 22 microliters, you might expect to have some inhibition. When we are doing the amplification, we might say, oh, there was a sample that is negative. Obviously, we follow quality assurance and quality control to determine that a reaction is valid. So we have to have a positive control, we have to have a negative control, and then we know that our reagents are working. And then we drop the digital PCR. So then we have all these reactions in 20,000 droplets. So then at that point, each individual droplet is a reaction. So then at the end, those droplets that have inhibition or that have inhibitory component, they are not going to amplify. Those that do not have the inhibitory components are going to amplify. Then we quantify them, and then we say, well, we have here 10 genome copies per microliter. Initially, what happened is that when you quantify these droplets, you have 20,000 droplets from 22 microliters. Now, out of those 20,000 droplets, the machine can say, oh, you have 10 out of 20,000 positive, and that's a good number. At least 6 of those droplets have to be, 6 out of the 20,000 have to be positive in order for you to say that a sample is positive. So then at this point, we have 2 major assays. One is for N1, and then the other one is for N2. N1 and N2 are genome sequences of the nucleocapsid protein that I showed you before here. This is the nucleocapsid protein. So then obviously it's a structural protein or sequence that is required, because it's like, just say we need arms, right? We need our arms, our legs. So then the virus needs that too. So it's a structural component of the virus. So then those targets for detections were based on N1 and N2. And then both for RT-qPCR and for digital PCR. So when we started doing this, we determined different numbers of genome copies in the sewage. So then based on that, we say, okay, if we determine this, what is called level of concern. That was important for the University of Arizona reopening and then keeping the university open in a safe way. Because when we went to dormitories, we were looking at these concentrations. So based on our wastewater-based epidemiology or wastewater surveillance of cycloronavirus, we said, okay, if we don't detect the virus, because we follow all the procedures with all the QA/QC guidelines, we say there is no action to be taken. So no problem, so there is no disease. Now, when we have a low concentration of N1 and N2, we have between 10 to the 1 and 10 to the 2, oh, we need to take these actions. And that's part of the epidemiology. And then the task force group at the University of Arizona COVID-19 task force group decided that was really key. when we have a moderate concentration between 10 to the 3, 10 to the 4, oh, we're going to be doing 10% random testing of the students here. And this is really important because the first time that we did this, when the university opened, we did a background study. And then prior to classes, we tested the sewage from those locations where sewage was produced, because there were no students, there was no sewage. But we collected samples from specific dormitories. We didn't detect the virus. Now, August 24, the university started classes. The students who tested positive were isolated. The students that tested negative were allowed to go to dormitories. Now, we conducted this surveillance, and then we observed that a day, let's say here you have August 24. So we have basically no samples because there was no flow at that particular dormitory. This was a case study. This was a specific dormitory. We've already published this information. This was posted in websites. We received a lot of congratulations from colleagues from all over the world. Science also made a remark about this. This is important because it's a research effort of many people at the university. And then that was really key. And then what happened is that on August 25, we detected the first signal. And then we detected this number, 1.61 10 to the 5. So that night, the president, the director of the West Center, and then the task force group met, and they decided that, oh, we need to test all the students in this dormitory. All the students were tested, 300 students, only 3, and then 2 were asymptomatic. They were excreting the virus. So these students were removed. And as you can see that on August 27, August 28, the sewage samples were negative. So that was key because at that point, out of 300 students who were tested, 3 were positive, they were removed. So that was important to avert an outbreak of COVID-19. And after that, based on the surveillance, that was also possible to do. So many institutions in the US follow these guidelines, and how wastewater-based epidemiology along with clinical testing was key in order to ensure the safety of the employees going to campus or the students coming back to classes.
Dr. Betancourt, to have some questions, if you could wrap up in about 5 minutes, then we can have some time for questions.
Yes, I'm gonna finish this soon.
Okay. Thank you.
Yes, wanted to say here that obviously our results have indicated that out of the 2 techniques that we are working on, RT-qPCR and digital PCR is more sensitive, and then we have been able to capture samples that were probably called negative by RT-PCR, and then drop the digital PCR is showing us. So capabilities to study the virus, to conduct wastewater-based epidemiology are available. This is going to be really important. And then I open this session to questions.
If you could stop your screen share, then we can see people's faces.
Yeah.
And Steve, I think there's one question in chat already.
Sure. Yeah, we have one from a couple in chat here. One from Jeff. He asks, Doctor, can you compare immunity levels of people who have had the 2 shots of Pfizer or Moderna versus survivors of the infection?
Well, I'm not a clinical, but I can say that both Moderna, from what I know, have 95% efficiency. And then Pfizer, I think it has 98% efficiency. So it means that, let's say, if we talk about, let's say, poliovirus vaccine. So 1 out of 1 million can get infected, with the vaccine. With this vaccine, which is actually messenger RNA, so what you are actually is stimulating your body to create, the immune response. I understand it's 95% is kind of high, but there is still a 5% probability of getting, not the immune necessary to avoid exposure. I mean, to minimize the exposure of the SARS coronavirus or to getting the disease.
All righty. And are the same wastewater tests being conducted at ASU?
Yes, actually, not only ASU, there are too many institutions in the US doing the same thing.
All righty. Dick Carlson, you had a question. You've got your hand raised. You want to unmute and take it away?
Yeah, this is a very interesting technique, and I've been watching it. It's really a great way for a campus. My question is about the scale. You're doing you're sampling groups of basically 300 people. I assume it would be overwhelmed if you did a whole community. If you went down to the central waste treatment plant and sampled that, you'd always get positive. How big a group can you handle to get useful results? Or
you can handle 1 million individuals, a facility serving 1 million individuals. So now, remember, when we talk about a sewage treatment plant, we receive the sewage that is received by the sewage treatment plant might have not only residential inputs, but can also have industrial inputs. So at that point, you might have a dilution component there that you might need to take into account. When we go to dormitories, we have a selected population of 300 individuals, more concentrated, and then we can detect the virus more efficiently. But no matter that, let's say wastewater from Agua Nueva, we are capable of detecting the virus at a level that we can determine if it's high or low or moderate incidence of a COVID-19 in the population.
So could you apply this to just part of the community to see if some parts of the community have much higher infection rates and is that possible?
Definitely. So that's the principle of wastewater-based epidemiology, just surveillance of the SARS coronavirus genetic material to determine the incidence of the disease. Remember, COVID-19 is the disease, right? SARS coronavirus 2 is the virus. So what you are targeting is the genetic material of the virus, because if the disease is present, we excrete viruses, we are infected with viruses, we are probably asymptomatic. This is a pathogenic virus. So it's present only when there is a disease.
All right, great. Thanks, Dick. We had a question in chat from Bob Delaney, and he asks whether this testing is being done at nursing homes where the population is more vulnerable.
What I understand is yes. And actually, we conducted some testing in hospitals, during, I think it was June, July 2020. But nursing homes, we have not tested nursing homes yet. We were contacted and then we were told that we're going to be receiving those samples. We never received those samples. So we have not done any surveillance in nursing homes.
Okie doke. One more question here in chat. Any significant outbreaks at the UA since the beginning of the testing?
The beginning of the testing was key. There was an outbreak and then that was averted. That was the case study that I presented. So then the virus was detected at that point, at August 25th. Then the reason was, we need to test every individual in this dormitory. The 2 individuals who were infected were removed. And then in that case, the samples came negative after that. And then there were no cases of the disease after that.
Okay. Going once, going twice, going 3 times. Okay, I think that's it, President Paul.
Thank you. Well, I'd like to thank you, Dr. Betancourt, for the information. Some of that got in, I know, over my head, but you know what? All those little tiny things to go look at, but I commend you and your colleagues for going through and coming up with a means and methods to be able to try to chase this stuff out and actually be able to identify stuff. So it's good for everybody to kind of follow and watch along, because it works, and that's the big thing. So
Exactly. Yeah, no, and I really thank you, everyone here, for your attention. I get really excited when I'm talking about something that is my area of expertise, and time doesn't matter to me, but I guess everyone is not the same. So.
No, and then the other thing, you hit a chord too. Rotary's real big into polio and polio prevention and vaccination. So what we do for all our speakers is we donate funds to be able to have 10 folks inoculated against polio, because it's still out there just in a few places, but it'd be nice to kind of get rid of that off the world stage. So
Yeah.
for your presentation. And then you did hit a chord when you hit the polio thing, because we all understand what's going on with that. So
Thank you. It's an honor to be here, and I appreciate your time. And then was really when I was invited, I say immediately yes. I am fascinated with this, and then altruism of people helping others.
again, thank you for that. So you're welcome to stick around. We got a few more things to wrap up, but
I have to go because I have another meeting coming soon. I have a symposium with another university, Michigan State University or University of Arizona. We have symposiums on Friday, but okay. I am very happy and then open to another meeting, probably make it shorter and then more focus. Yeah. But it was great to meet everyone. My aunt is also part of the Rotary. She's a gynecologist. And then I'm going to talk to her and say that she's not here. She's back in Venezuela, but yeah. She's part of the Rotary, a member too. Yeah. And I think it's an honor to have spoken to you all today.
Well, again, thank you. I know we all appreciate it. We all learned something out of this. So yeah, chasing this awful monster around. So thank you.
Yeah, appreciate stay safe and my blessings to everyone.
Yes. Okay.
Bye.
Bye. A little bit of time, not much though. Any happy bucks that are out there? Happy bucks going once. Okie dokie. Don't see any hands up, so I guess nobody's happy. I was gonna say today, if you remember, yeah, it was declared a pandemic a year ago. I think yesterday. The NCAA tournament was canceled a year ago today, and I just found out my Kansas Jayhawks are COVID bound, so they are not participating. I don't know if they'll make the tournament, and if they miss, that'll be it's not good news. My son sent me, so I'm not happy. But Arizona's not playing either, so I don't have either one of them to watch. So a couple other things. Today is National Girl Scout Day. More importantly, it's National Baked Scallops Day. So go for that one. And here's something for everybody. Right now, we're on Mountain Time. Come Sunday, we'll be on California time. Daylight savings time. We don't change, but the world evolves and changes. So if you have somebody on the East Coast, 3 hours difference instead of 2. So when Mr. Rich comes back to visit, he's got to remember it's 3 hours different. And Carol, I saw you, so you'll change. You're gonna have an hour change going forward. And then I think if Dick's over on the West Coast time, so we'll be the same time you are for next week, Dick. But remember those things. Any other comments good for the club? Anybody want to have thoughts?
Paul, I just wanted to say we're looking at April 10th for a spring road cleanup, and there'll be more information confirming that. But you know, maybe just circle that on your Saturday morning calendar that we will be having the road cleanup on Saturday, April 10th. More to follow.
And just as an FYI, I know Finney's is open for breakfast on Saturdays too, so maybe we'll do something socially distanced. It's fair. I've had a couple meals up there during the day, so that might work out. And so that's coming up.
President Paul,
yes.
I wonder if Jimmy has anything he wants to share with us, if he's still there, from his pets experience.
Yeah, I could certainly do that. I gave a little bit of a roundup to the board on Tuesday during our meeting, but yeah, off and running. It was really impressive. Rotary put together, the district put together a really good presentation. Imagine being on Zoom for really a whole day, which you're probably just gagging just thinking about that. But it actually, they pulled it off really well. They had a really good presenters. They had a very, very active engagement, I thought, in the different sessions. So for those that might not know, PETS is the acronym for President-elect Training Seminar. And it's basically a way for me to get ready for next year. And so they kind of have some how-tos and collaborations with other Rotarians that are in the president-elect position. And so not knowing what to expect, kind of prepared for the worst and hope for the best type of thing. And I was really pleasantly surprised by everything. That was outstanding. And yeah, so I guess it's time for me to start implementing some of those things and start getting ready for next year. My apologies in advance. Paul's got big shoes there to fill. So yeah, we'll stay tuned. But it was a neat event, and yeah, definitely pass that feedback on to the district. I mean, I thought it was really well done.
Great.
Couple other things, the district peace monthly meeting is on Monday night. That will be included in the wagon wheel that'll go out on Monday. There's also going to be an additional Rotary Leadership Institute later, I think in April or May. I'll put that information in the wagon wheel if people would like to participate. Some of the newer members or those people going into on the board or whatever may want to attend some of that going forward. So those things are coming up. A couple other things. We'll have a familiar face next week if he doesn't get snowed in. Oh, never mind. That's Merle. He can get snowed in and give it to us, but he's going to talk about American agriculture and what's going on these days, I believe. And then on the 26th, we're going to hear from Elizabeth Slater, who's with Yoto on their strategic plan and what's going on with that. So I think that'll be real good. If you all remember, you know, we've been kind of going through things and helping out in different places. Yoto, we did quite a bit at the beginning of the year. We've done with Erickson, the reading things, and, you know, it's kind of, we all are able to kind of reach back and kind of help out where help can. We didn't have, you know, I kind of missed the rodeo. The virtual rodeo just didn't do quite the same thing. The Bucking Broncos just were not as exciting as previous years, but I'm sure we'll be able to get back into that next year. But we have been able to help out the community in several ways. And so we'll keep going after it. And hopefully one of these days we'll all be back together, combination virtual and in person, because I think that's where the direction will go. That allow people to, when they feel comfortable, show up. But until that time, still be included in virtual sessions. As a wrap up, just an anonymous saying, but something to think about. I mean, our lives have all changed in the past year, and it's been a little bit of a little stress and such, but today is only one day in all the days that will ever be. But what you do today will impact everything that happens in all the days that will ever be. Choose wisely, be positive, be hopeful, be helpful. We are and we'll get through this. Have a good week. Be safe. If you haven't got your shot, chase it out a little bit more. The county seems to be opening up a little bit more. So I think we're going to get there. And also there'll be a survey out to the club members probably tomorrow sometime, now that I don't have a basketball game to watch. So, and we'll see how things go forward. Be safe. See you next week.
Take care.